Okay, we're live for section 9 and 11. If you are on YouTube, a little bit early, we're only about 10 seconds into this filming, so type in your name and say here or whatever, so I can get a record of who's present with us today. Good morning, Jessica. How are you? Can you hear me, Jessica? Good. I'm glad you're here. Okay. We'll let some other folks get in, and then we'll start this lecture on chapter 10. Joyce, good to hear. Good to see you. Can't hear you, but it's good to see you. Got 30 of you, so it's going to take a minute to get the roll. Let's see, Riley. Good morning, Riley. Good morning, Landrea, and also Millette. Good to see you. I should say hear you, or at least see your name. Hi, Jaquita. Glad you're here today. Everybody seems to be healthy at this point. It's going to take a little bit of time with this roll because we got about 30 people if they show up. Tiffany, good morning. Tiffany, have we gotten into chapter 10 yet in terms of muscles? Okay, you go ahead and do that. That's fine. Okay, good. We haven't gotten into chapter 10 yet. Okay, Amanda Rouse. Good morning, Amanda. I hope you guys had a nice spring break. Sure was nice for me. Yes, I've got you checked as present. Sure do, Turquita. Hello? Hey, yeah, we're online. You got a problem over there? When you, in the search bar up there, did you type in John Pritchett? Okay, up there in that search bar, you put John Pritchett and also type live and see what happens. See if you don't get a picture of me sitting here with the phone to my ear. Hmm. 10. Mm-hmm. Uh-huh. Could be that. This one is live, and I've entitled it, same thing, Chapter 10, Muscle Structure, and I think it's /function. So you should be able to see me on the camera if you're in that area. We're about, I don't see any, let's see here. We're about 8 minutes into it, so I'm just waiting for folks to come in. I'm probably going to have Jaquita doesn't see me. Well, how about that? Well, I've got kids that are, I see Shay and Lauren are in. Now those, yeah, Chantel and, well, I don't know what to tell you. We haven't had any problem on the last 3 presentations. Okay, what did you do? That's right. Okay. All right. All right. I'm going to mark you here. We're going to start talking in a few minutes. Okay. Bye-bye. Oh, me. Okay. Good morning, Nicholas. And Chantel. Here we go. Number 29. Yeah. Monica. Okay. Good to hear from you, Monica. Okay, Victoria Fernanda. Is this the same Victoria Casper? Tyandra, I see you're here. Good. Okay, I see this Alexis. Okay, Alexis. Yeah, I've got you. That's the first time I called your name. You're right, Alexis. So let me mark you present. Phantom of the Popular. Nobby. Okay, Nobby. Thank you. Never would have known who that was. Okay, I see I've got, let's see. What did I see that? Shay and Lauren. Oh, Chantel here. Okay, I see. Is that two people? Shay, I don't know of a Lauren in my class. Tiffany, thank you. You're back. Okay. Okay, I'm missing Bryant. I'm missing Kayla. I know Jalen's getting his teeth worked on today. Asada. This is one cumbersome way to do things, I can tell you that. And Hannah. Don't see Hannah. And Breely. I don't see Breely there. And don't see Ramsey. Okay. Haley. Haley, are you there? I don't see your name. Don't see Brandy. And Caitlin. Got Amanda here. She's present. Okay. Well, let's get started on this, on chapter 10. So let's look on page 340, if you would, in your textbook. For page 340, and look down, if you would, at the bottom of page 340, and you see the three types of muscle. So as you look at that, what I want you to do is, see what I do with that paper. So look at figure 10.1 and name three physical differences between skeletal and cardiac muscle. Three differences between skeletal and cardiac muscle. And somebody's calling me. Let me see who that is. Hello, I'm in the middle of a lecture. Okay, listen to me. Listen to me. Are you on the internet? Okay, have you typed in YouTube? And now, John, how did did you put Pritchard or Pritchett? Excuse me, folks. Got a little problem here. What does your search bar say? Okay, and you clicked on that, and you should come down. And what you want to do is just scroll down until you find John Pritchett. It doesn't have anything by it besides that big orange. Yes. Got a J in the middle of it. Click on it. Can you see me waving in a screen? Okay, go to chapter 10, muscle structure and slash. It says structure / function. Got it? You can see me waving at you. Good, okay. I'm going to start talking, all right? We're in chapter 10 on page 340. Okay? Bye-bye. I can understand the frustration. I have the same thing. Okay, so I ask you to look at the figure down there, figure 10.1, and give me 3 differences between skeletal muscle and cardiac muscle. As soon as you get it, type it in. Whoever gets there first. Joyce says in probably an older edition, it might be on page 338 in some of the books. So those of you, if you have an older edition, look on page 338. If you got the new one, look on 340. A couple of pages difference. Thanks, Joyce. Who is funny? But Teresa Tyndall. That's Funny Balloon, right? Okay. Okay, now listen to the question. 3 differences in structure. Okay, Jerkita says there are branches in cardiac, and so therefore I assume, Jerkita, that you're saying that skeletal muscle doesn't branch. So you want to be complete with that, okay? Okay, now, Joyce, cardiac, that's not a structure, that is a function, okay? Structural differences, looking at the picture down below. Okay, Alexis, that's good. Skeletal muscle is multinucleated, whereas cardiac only has 1. And sometimes it says it has 2 nuclei, but we'll just stick with 1 there. Okay, Victoria, skeletal is long. It's sort of cylindrical. That's correct. Whereas you have branches in cardiac. Good. One more feature, one more structural feature looking at the picture. Can you see one more difference? One more structural difference. This is good we're doing this because you're giving some answers, but they're not associated with structure. They're associated with function. So you want to make sure you read that question carefully. Skeletal muscle is long and cylindrical. Cardiac is short and wide. Good. One more structural feature. Right, that's correct. All right, now both of them have striations, Ramsey, so that would not be a difference. They're both striated muscles. But Jerkita, you see what's different there? That third characteristic, third structural difference is the intercalated disc. So you might see questions like that on the next test. And if you put down involuntary or voluntary or whatever, that's not a structure, that's a function. So you want to make sure you read the question carefully and understand what it's asking for. That way you'll score well, which is what I want you to do. So we're going to look at skeletal muscle today. And so I want you to turn over to actually page 345. And Joyce, if it's not on 345, this illustration might be a couple of pages off. You can type that in and everybody can see that. But I'm on page 345. So as you look on page 345, you look up at the top and it says the big picture, doesn't it? And then it says right under the big picture, in very small print, it says figure 10.6. So let's look at the structure of skeletal muscle. And you see, as you look at the bone on the left-hand side, the upper portion of that illustration, then you see tendon, and then you see this kind of a shiny material coming off, and you look right below the muscle, it says epimysium. All right, that's a type of connective tissue. So the outer part of the muscle is covered by what we call epimysium. And you see it's where the black line is pointing from epimysium. You see they pull it away from the muscle. We usually don't try and eat that because it's kind of tough. But then notice as you move into the cut face, that flat face looks like it's got a number of, let's see, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, got 11 or 12 structures in there. Can you see the little gray area around each of those structures? And you see that if you come out a little bit from the face, you see the word perimysium. From that cut surface, you see perimysium. That's the same kind of connective tissue that epimysium is. And so you see the perimysium surrounds these little red sections, and you have one section that extends from that cut face, and you see the word fascicle. And as you look at the fascicle, you see it's covered. That's that perimysium. You see you got a little, where perimysium, the word perimysium is. You see there's a line going to the left pointing to that gray, slender gray material that surrounds a bundle. We'll use that. That's actually a fascicle. And then you see going up from perimysium, it points to that gray sheath around the fascicle. Now, as you look at the cut face of the fascicle, then you see there are some other structures within the perimysium. And you see one of those structures extending from the cut face of the fascicle. Got a little bit of red and blue in there. Now, as you look at that extended structure, you see there's an arrow that points down. And that's right beside the word muscle fiber. So what is extended there, that view shows you that you have a muscle fiber extending from that fascicle. So you got 1, 2, 3, 4, 5, 6, 7 muscle fibers in that fascicle, and one of those is extending to the right. Now, right above that muscle fiber, I want you to come back up to where you see the fascicle, and you see that extended muscle fiber coming from the fascicle. Right above that muscle fiber, well, right above that extended structure there, I want you to write muscle fiber. And then right above that, I want you to write muscle cell. They are the same thing, but people use different terms to describe them. So that extended structure coming out of the fascicle is a muscle fiber. It's also a muscle cell. And I want you to write the word myocyte. Myocyte also right above muscle cell. So you have MYOCYTE. 3 different names for the same thing. Now, the muscle fiber, the muscle cell, the myocyte, also has connective tissue surrounding it. I want you to come down to where you see that bracket and it says muscle fiber. Now look to the left of the muscle fiber and you see the word endomysium. That's the same connective tissue as the perimysium and the same tissue as the epimysium. So you've got this connective tissue around the muscle cell or the muscle fiber. You've got the same connective tissue around the fascicle, which is a group of muscle fibers. And then you see the epimysium that surrounds all the fascicles that make up the whole muscle. Now, all 3 of those tissues, the endomysium, the perimysium, and the epimysium, all come together, go back up to the top, where you see the large muscle and the bone and the tendon. All 3 of those connective tissues extend past the muscle to become that white structure that we call a tendon. So you see the muscle is well anchored, very well anchored to the bone. And of course, that's the purpose of a tendon. If you break that, cut that tendon, break it or whatever, then you can contract the muscle as much as you want, but you won't get any movement because it's not attached to a bone. Now, what kind of connective tissue is the endomysium, the perimysium, and the epimysium? What type of connective tissue is it? That's correct, Drakita. It's dense, regular connective tissue. Remember, all the fibers are all parallel to each other. Very strong collagen fibers that not only support the muscle, but connect the muscle to the bones. So up there in that first illustration, when you move to the right from the tendon out to the muscle, out to the fascicle, out to where you labeled that extended structure, which we call a muscle fiber, a muscle cell, a myocyte, you can actually put a little note there and say, covered by endomysium. Now we're still looking on page 345. Come down to that middle section where it is describing the muscle fiber according to their verbiage. And you come over to the left and you see that term endomysium again. And you see it's sort of a grayish type of tissue. So move to your right and you'll see where a tissue has been cut away, but they label that tissue the sarcolemma. Now the sarcolemma is the same thing as a cell membrane. That is the cell membrane of a muscle cell. So what is that cell membrane made of? Turquita, you're correct. It's a phospholipid bilayer. So it's going to look just like regular cell. We're going to find out in a few minutes, though, that it's got a little difference about it. Now, before we go find that out, I want you to do this. As you look along that muscle cell, starting with the endomysium, and you move toward the sarcolemma, which is the normal cell membrane for a muscle cell, go all the way to the right, and you see this extension, another extension, coming from the cell. You see the cut face of the cell. And you've got one looks like a log, this cylindrical structure. Above that cylindrical structure that's got those little bands in it, I want you to write the word myofibril. It's down here right below the cell, and you need to see how to spell it. Myofibril. Just reinforcing this for you. Oh, Sheldrick, that's you, Rhonda. Okay, I didn't recognize that. Thought maybe somebody from USC or Clemson was looking in on this. All right, you've got that labeled myofibril. Now come down just under that myofibril, and I want you to write the word organelle. Organelle. Andrew, you want me to repeat about the myofibril, that little extension that's coming from the cell? You see the little bands, some light bands and little tiny blue bands in the middle. That one cylinder, I want you to label it Myofibril. That's the same thing. You see that big red arrow going down. But just write it right above there just so you can get it straight in your head. And then I want you to, underneath what you just labeled as a myofibril, I want you to call it, list it as an organelle. Now, you know, going back to chapter 3, when we entered into the components of a cell, the structures inside were called organelles. You had those structures. Well, you tell me, somebody tell me, what are the structures, the organelles that you find in a cell? Good, Tiffany, that's right. Golgi apparatus. Okay, somebody else, give me another organelle typically found within a cell. Okay, Landrea, good. Mitochondria. What's another one? Funny balloon. Okay, that's the SER. That's the smooth endoplasmic reticulum. Good. Monica's got the rough. Okay. Alexis has got mitochondrial lysosomes. Very good. And Alexis, you got nucleus. So you remember those are components of a cell. So this myofibril is an organelle only found in muscle. And of course, for us, it's going to be the skeletal muscle or the cardiac muscle. So make sure you understand those structures that look like little logs stacked on top of each other. Those are organelles. You don't find them in our liver cells. You don't find them in our brain cells or anything like that. Now, let's go back to page 343. It's actually going to be figure 10.4, and hopefully that'll narrow it down for you. So you're seeing something that's very similar. So as you look at the top, you see it's almost identical, isn't it? It's just a little different size there. You see the bone, you see the tendon, which is an extension of the epimysium, the perimysium, and the endomysium. You see the fascicle. And then you see the muscle fiber extending from the fascicle. So that fascicle, if somebody says, what's a fascicle? You simply say it's a collection of muscle cells, a group of muscle cells. Look on page 343. Make sure we have the right thing. Up there at the top, it says 10.4 for me, but if you're on 343 and you're seeing the bone up at the top, little smaller version, smaller picture of the bone, the tendon, the muscle, and then you see the fascicle coming out of the muscle, and then you see the muscle fiber. Looks like you might be in the right place. Okay, Joyce says it might be on 341 on some of yours. There is a picture there. I guess it's just the different versions, or the different editions, rather. Now, on top of this muscle cell that I want you to look at, you should see from the left-hand side the top of the muscle cell, you should see endomysium, sarcoplasmic reticulum, transverse tubule, terminal cisternae. You keep moving to the right and you see sarcolemma. So if you're seeing that, then you're seeing what I want you to look at. So as we look at that, you can see endomysium, that's that dense connective tissue that encircles every cell, muscle cell. And if you look over far to the right, you can see them lifting up as it were a sheet of paper or something like that. You see the sarcolemma. And what we want to do is we want to look inside the muscle cell. You've already got those myofibrils marked out. Now they're going to point to myofilaments. Don't worry about that at this point. We're not there. We're looking at those log-shaped structures, and those are myofibrils. If you go to the right and you see that extended myofibril, it's extended past the cut face, and you look right below it, you see myofibrils. It's got a little ring drawn around it from myofibrils. Those are your organelles. That's the last thing we talked about. Those are organelles. So now we're looking inside the cell. And right below, as you look to the right of that muscle cell, you see myofibrils. That's the one that's extending past the cut face. And look at the term below it. And you see cytosol, don't you? Could be cytoplasm. Either one would be acceptable. It's intracellular fluid. That's what these organelles float in. We call it cytoplasm, call it cytosol. Now, beside the word cytosol, C-Y-T-O-S-O-L, that's correct. That's correct, Rhonda. Is it Rhonda? No, it's Teresa. I think it is. Sheldrick. Anyway, myofibrils are organelles. That's correct. Only your muscles, cardiac and your skeletal muscles, have those organelles. And you see below there is cytosol. I want you to write down the word glycogen. Okay, Rhonda, I'll get it straight after a while. You're Sheldrick. Okay. Who can tell me what glycogen is? Somebody asked you a question for that. What is glycogen? That's correct, Monica. It's a complex carbohydrate made up of a number of glucose molecules put into sort of a chain type thing, and it also has some branching chains. What do we use glucose for? What is its purpose? Alexis, you're correct. Georgina, you're correct. Monica, you're correct. Ramsey, it's not bodily tissue. It's actually a substance that we use. Monica says it's energy, and Alexis says that. Tyandra says that. That's exactly right. We store some glycogen in our muscle cells. It floats around in the cytosol. So it is an energy molecule for us. We have a certain storage capacity for glycogen. When we get out there and start playing softball, soccer, or something like that, basketball, then we start using up some of that glycogen. So I want you to know that that substance, that carbohydrate, is in the cytosol. Now, right where you see cytosol and you put a little arrow maybe out there beside it, you have the word glycogen. That's correct, Joyce. You're right. And Ramsey, now you got it down for energy, and that's good. It's used for energy. I want you to write the word myoglobin. 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 Of course, myo is a root word for muscle. This is a protein that carries oxygen. So we have a little bit of a storehouse of oxygen in our muscle cells. What's its purpose? We said it was to store oxygen. Now, you know there are some creatures, some of you probably watch some of the National Geographic channels and so forth. And so, let's see, Monica says, does myoglobin interact with hemoglobin to transfer oxygen throughout the muscle? I'd have to look that up. That's a good question, Monica. And I'll get it back to you. Alexis says it's similar. Ramsey says yes. Anyway, I'll look it up. Now, there are some creatures that you know about, like whales and sea lions and so forth, seals and sea lions. They have a greater amount of myoglobin, which allows them to dive deeply in the water and stay underwater more than 3 or 4 minutes like you and I would do. So they can stay down there quite some time, whatever they do at that particular depth, 800 feet or something like that, and they still have plenty of oxygen. And they come back up and they do some breathing. Must be a pretty efficient system in order to do that. But we have some of that also, but we're not going to go down and stay down 20 minutes underwater. But they can do that apparently pretty easily because they have more myoglobin than we do. Now, let's look back at the picture of the muscle cell. And as you work from the left-hand side, you see endomysium, and you come to this structure called Sarcoplasmic Reticulum. That's the blue structure. I assume you've got a colored version there, and so you can see the kind of I don't know what kind of blue it is, whatever you want to call it. But it's like the endoplasmic reticulum that you would find in a typical cell. Now you move over to the right from sarcoplasmic reticulum, and you can circle or check or highlight, however you're doing it, the word transverse tubule. These are extensions of the sarcolemma. You can see the line pointing out one of those sort of light brown cells, tubules, light brown tubules. They try to show you over on the left where it says opening of T tubules. T tubules, the same thing as transverse. And you see that they go through the muscle, and they're closely associated with the sarcoplasmic reticulum. Now you come to the next term and you see the word terminal cisternae. That's part of the sarcoplasmic reticulum. T-tubule, the transfer tubule, is associated with that section. It's sort of an inflated area, a little enlarged area, as opposed to being tubular, as in other parts of the sarcoplasmic reticulum. Now, come to the bottom of the cell, and you see, of course, a block with a little blue arrow pointing down and to the right. And now you see the block that has what's called a triad. And that means it's consists of three structures. Two parts of it are the terminal cisternae. That's somewhat inflated, enlarged, a section that's not so tubular. That may be a new word to you to understand cisternae. That's a Latin word. When you see A or E on the end of it, that simply means there's more than one. Cisterna would be singular. But some of you may have heard of the word called cisterns. C-I-S-T-E-R-N for singular and add an S for plural. People for thousands of years have dug holes in the ground, or maybe not even dug holes in the ground, but some have done that, and they have built some sort of a container. Some people have used bricks. Bricks are made out of mud and so forth. And so they would build this big hollow cavity in the ground, and then they would build a cover over it. So when rainwater came and it came off their roof, it would go into that container, which they call a cistern. From there, that people would have drinking water during a dry spell, bathing water, cooking water. So that's the way they conserved water if they didn't dig a well and have a source 40, 50, 60 feet below the surface of the earth. So a cistern is an enlarged area, in this case, of the sarcoplasmic reticulum. Let's see. Let me answer your questions here. Riley says, did you say the T-tubule were associated with the Sarcolemma? Yes, they come from the Sarcolemma. The T-tubule is an invagination. It's where it points down or away from the surface, and it's forming a cavity, a small cavity. That's what that little T-tubule is. We understand a tube. When we think of it, we think of something that's round and hollow on the inside. And so that tubule could be considered an invagination. That's an infolding of the tissue. And in this case, that invagination becomes the T-tubule. So yes, Riley, the T-tubule is associated with the sarcolemma as well as with the sarcoplasmic reticulum. You okay with that too, Nabi? Andrea, you okay with it also? Good. Okay. Good deal. Okay. So that T-tubule lies on top of those two terminal cisternae. Now, you can see that picture down at the bottom right-hand side of figure 10.4, and you can actually see sarcolemma. That's the cell membrane. You see where it invaginates. You see the hole there. and it forms a tubule. So you see the little opening of the tubule. Now, that has a functional property to it, and we're going to look at it. Maybe probably won't get it done today, but we'll come close to it. Everybody okay with that? So let's go over to page 345 again, where we have the larger picture of the bone, the tendon, the muscle, the fascicle, and so forth. That first page that we were on. Look on 345. And you see the myofibril. Now, look at the myofibril on page 345. And you see the kind of dark area, we would call it, the sort of reddish area, and then you see sort of pinkish area, don't you? And then you see a nice big middle portion that is red, and then you see the pink area on the other side. Now, as you look at that picture, you see where the little zigzag line on the left is? And there's a zigzag line. That's why they call it a Z line, or sometimes they call it a Z disc. You see where those are? Okay, now look down at the bottom picture. And again, you see the zigzagging line. Everybody think is okay with that. You see one on the left, you see one on the right. Now look at when the arrow comes down from the myofibril, look at the word that is used to the left of the arrow. And you see the word is myofilaments. Myofilaments. And these are actually the proteins which are involved in contraction. So when you look at that word myofilaments, I want you to think those are proteins. Now, how do proteins come into existence? What process? What event occurs in order to produce these protein filaments that we're going to look at, the thick and the thin and the elastic? That's correct, Jerkita. It's that event called protein synthesis. So transcription has to occur, translation has to occur. That's correct, Mallory. Those two events result in the producing at least of a polypeptide at that point. You remember when we talked about protein synthesis that the first thing we produce is a polypeptide. That's the first step. And the second step occurs, second, third, and fourth steps. Remember the secondary structure, tertiary, and so forth. That's when the polypeptide goes inside the endoplasmic reticulum, and some other enzymes work on it so we get the right shape. That's correct, Alexis. That's correct. That's right, Ramsey. That's correct. So what we're looking at here are actually 1, 2, 3, 4 protein filaments. Now, most of the time when we think of these myofilaments, we think of the thick filament and the thin filament. So where you see the Z line, that zigzagging line on the left and on the right, that forms a structure called the sarcomere. So you can draw a little line. You can label this from the one zigzagging line on the left to the other zigzagging line on the right. That is a sarcomere, and it is the contractile unit of the muscle. You're correct, Tyandra. You're correct. When we're looking at muscle, that's correct. The myofilament is a protein filament, and there are 4 different protein filaments that we're looking at as we look at this picture. Now, we have a tendency to just think of those, the big red ones and the little skinny blue ones, the thin filament and the thick filament. But there's more to it. There always is. You don't get the whole thing because you're not going to become a histologist or a cytologist. You're looking to become a physician's assistant or a nurse or a physician or whatever, and you usually don't get into all of that. But it's a beautiful arrangement, just beautiful how it works. So from one Z line to the next Z line, that's called a sarcomere, and it is the contractile unit of a muscle cell. Now look over on page 346. 346. Got just a few more minutes here and we'll close this out. Now on 346, I'm looking at figure 10.7, and it has a similar picture at the top, but it's labeled it as a sarcomere. So as you look at the enlargement of the sarcomere, as a matter of fact, if you look down at that picture at the bottom and look over to the right, you see written vertically, you see capital T, capital E, capital M, and then you see 38,000X. TEM means transmission. You don't have to know this, but it's transmission electron microscope. And so what you're seeing by that gray and black or whatever shade you want to call it, right beside that word TEM 38,000, that is an actual picture taken by a microscope of the sarcomere. These electron microscopes can go up to several million times in magnification. We can almost get up to the actual protein structure that makes, in this case, the muscle, composes the muscle. Amazing stuff in there. So as you look at that transmission electron microscope, you see it's been labeled. As you move to your left, you see, you come down the bottom of that illustration, you see I band, and you see and I means isotropic. Isotropic, light gets through there. You see the light area, you see the dark area, the Z disc, that's that Z line. See, that's a type of protein also. And you keep moving to the left, and you see down at the bottom, they call the dark area the A band, which is anisotropic. So it doesn't let as much light through, therefore they call it sometimes the dark band, but they call it dark because it's anisotropic without some light. And then, of course, you go to the next one, and you see an I band again. Some of you are having a hard time to find it. Joyce says that Picture's on 344. Different edition, but that's okay. It's probably the same thing. So you see from one Z disc, sometimes they call it the Z line, sometimes they call it the Z disc. From one Z disc to another, or one Z line to another, that is the contractile unit. Now, what we want to do in closing up the lecture is you want to go above to the illustration. Should be in sort of a bluish purple color and a light, what do you call it? Light red, dark red. My wife needs to be here. She knows all those colors. I don't know them, but anyway, you basically have showing here 1, 2, 3, 4, 5, about 5 different proteins. Now, what I want you to do is look at the word thick filament. When you see thick filament at the top of that rectangle down there, when you see thick filament, I want you to put the word myosin, M-Y-O-S-I-N. That's a protein. You look to the left and you see thin filament. And above that, you want to write A-C-T-I-N, actin. So when we think about the proteins that are involved in contraction, people typically think of actin and myosin. Are there other proteins? You bet there are. And we'll get to them a little bit later, but we're not going to do it today. Now, I want you to notice, you see where it says M line? In the very center of that illustration, got 3 blue lines going down through the kind of light orange or light brown, whatever, reddish brown. See M line? That's a type of protein too. It helps to anchor the myosin filaments. Now, one more protein I want you to be familiar with. Look at the first myosin filament. You see it looks like a little rod in the middle, and then it's real bumpy on both ends of it. Some folks might say, well, it's kind of like leaves on a tree or buds or whatever. But then if you move to the left, you see it looks like something like a little spring. And it's called an elastic filament. That's right, Joyce. Thin is actin. That's correct. And you see where it says elastic filament, looks like a little spring. I want you to write the word above elastic filament, T-I-T-I-N. T-I-T-I-N, Titan. Now don't put down T-I-T-A-N like you have these, the Titans that are in Greek mythology. That was a bunch of them false gods. And then you got cartoon characters that are called Titans and stuff like that. But that's an elastic filament. You see how it looks like a spring? It's got some elasticity to it, so you can stretch those muscles. Sometimes they stretch when you pick up some weight. They stretch a little bit. This gives the muscle a little bit of elasticity, but it also, you see it anchors. If you look in there, it's right in the Z line. It anchors the Z line to the tip of the next myosin. If you move to your left, you see another head of myosin. Or you can come down when you look at that first myosin filament, and on the left, you follow that spring and it's anchored into the Z line. So stuff's not just floating around in there. It's all organized in a particular fashion so that you can have these filaments slide over each other, producing movement that's useful. Do you have any questions at this point? That's right. That's right, Tiffany. That's right, Alexis. Ramsey, Titin. So they're not necessarily the contractile units, but they're part of the contractile unit. My office number, Nabi, is 5416. Anybody have any questions? It's a little more challenging, isn't it, to look at that picture and have me try to talk you through it than it is to have the screen up in front of us and I can put the green dot on it and so forth. Yes, as far as I know, Tiffany, it is. And Riley says we will watch again Tuesday at 9:30. That's the plan. And so I know when I end this thing, it saves the lecture. So I guess you can upload it on your computer. Let's see. Teresa says, when is our test? Not sure yet. They're looking at a couple of ways to get this accomplished, and it has not come down to me yet. So in the meantime, be studying chapters, what was it, 5 and 6, I believe it was, in terms of the skin and the bones. Keep studying that. Teach your closet door, your bathroom door, your chest of drawers or whatever, and get really good at it. You want to get it where you don't have to look at those notes. That's the whole thing there. Get it in your head. And then as soon as we get it worked out, we'll, in terms of testing, we'll move in that direction. But we got to keep going on with the information here. Let's see what else we've got in here. Joyce says, let me look at this. Let me go up a little bit more. I have to watch it again to get a clear understanding. That's right, Tyandra. I'm right there with you. I have to do the same thing. And that's what happens when you try to teach it to the closet door. You're getting it in your head. The more you talk it out, the more in order it gets. And so you can pretty soon see your notes in your mind, and then you can just pull those answers off. Now, let's see what else we got here. Okay, Jerkita, we don't know about the test yet. And the same for Shay. And in light of this coronavirus, we need a 100 on that exam we're missing. Yeah, that little booger's really creating some problems in the whole world, isn't it? It reminds us how we're here only for a while. Got to be ready to go. Tiffany says, great, my kids had 1 million questions on their work during this lecture. Oh, my. But you're a woman, Tiffany. You know that song says I can da-da-da-da-da-da do all this because I'm a woman, and you can multitask. You girls are good at that. Let's see. Riley says, not that I don't love your class, but being in my PJs drinking my coffee from my living room is kind of awesome. Well, I'm glad you're getting some joy out of this. And your hand, obviously, and your shoulder are working well so that you get your coffee in your mouth and you're enjoying that. Glad you can do that. Okay, Nabi, wait. Let me be more specific. Laugh out loud. What's the phone number to your office? It's 843 You got a pencil there, a pen, 843-661-8053. Okay, any other questions? All right, well, you guys, let's see. What's your entire office number? It's 5416. That's on the 4th floor. Don't all of you come up here. They don't want you up here because of social distancing and so forth. Keep your hands away from your face. I'm having to work on that myself. You see me sometimes scratching my nose or whatever. I've got to stop that. Don't want any transmission. Make it hard for the virus. Wash your hands well, and then keep them away from your mouth and your nose. Okay, they're good. That's right, Tiffany. That's my office number for the phone. Okay, any other question? I'm going to end this stream, and Lord willing, we'll see each other next Tuesday, and we'll talk about how the muscle contracts, how the message from your brain goes down to your hand or whatever, your foot, your leg, and muscles respond in a coordinated way, and you get where you need to get. All right. Have a good weekend, folks. See you later.